Background of the study. Prebiotics are non-digestible compounds that host microorganisms selectively, used to benefit the host's health. They are the food components for beneficial bacteria living in our bodies, called probiotics. Sargassobiotique is a first-of-its-kind prebiotic extract derived from the brown seaweed Sargassum polysystem. It is a dark brown powder that made possible through an innovative, enzyme-assisted extraction technique. Hence, the name Sargassobiotique. Enzyme-assisted extraction is a green technology and an environmentally friendly extraction technique. Problem statement and objectives. Commercial prebiotics such as inulin and fructooligosachous are mostly obtained from plants. They are seldom derived from marine sources such as seaweed. Ecologically, the utilization of seaweed as a source of commercial prebiotics offers unparalleled advantages. It does not compete on land use for food production. Seaweeds have fast growth rates and do not require arable land, fresh water, or even fertilizer when compared with terrestrial plants. Extraction process. First, Sargassum polysystem was dispersed in pH-adjusted distilled water. After the addition of enzyme to the suspension, the extraction was carried out inside an orbital shaker at predetermined incubation temperature overnight. Once the extraction is complete, the suspension was boiled to inactivate the enzyme. The suspension was centrifuged to collect the supernatant. The supernatant collected was precipitated with ethanol overnight. Finally, the sample was centrifuged to separate the pellet polysaccharide fraction from the supernatant. The pellet was vacuum oven dried until constant weight and thereafter kept at -20 degrees Celsius until further use. Evidence-based data. Soy-based yogurt made with Sargassobiotique. Sargassobiotique has been investigated as a functional ingredient in soy-based yogurts in our laboratories. Our laboratory studies have shown that Sargassobiotique can be successfully incorporated into soy-based yogurts, as it does not affect the fermentation process of soy-based yogurts, and it supports the growth of Lactobacillus bulgaricus and Streptococcus thermophilus. Fourier transform infrared FTIR result. FTIR was used to further characterize the polysaccharides in the extract. A strong peak around 3,200 per centimeters was observed, which corresponds to hydroxyl stretching vibrations, showing hydrogen bonding between hydroxyl groups in the polysaccharide backbone. At 1635 per centimeter and 1416 per centimeters, signals linked to carboxylate groups were detected, confirming the presence of alginate, a uronic acid-rich polysaccharide. Finally, a peak at about 1038 per centimeters indicates guluronic acid in the extract. Brine shrimp lethality assay. In vitro cytotoxicity studies also found that the prebiotic extract was non-toxic. The extract of Sargassum polysystem showed no significant toxicity to the Artemia nauplii, with an LC50 value of 2,281 micrograms per milliliter after 24 hours of exposure. LC50 values greater than 1,000 micrograms per milliliter are considered to be non-toxic. Hence, this indicates that the extract is non-toxic at the tested concentration and may have potential as a functional food ingredient. Benefits of Sargassobiotique. Sargassobiotique is a functional ingredient that supports digestive health. It is targeted towards individuals who are health conscious and interested in maintaining their digestive health. The cosmetics and skincare sectors can potentially benefit from Sargassobiotique. Sustainable Development Goals (SDGs). This project aligns with several SDGs. First, SDG 3, Good Health and Well-being. The prebiotic extract supports the growth of beneficial microorganisms in the digestive system, which promotes gut health and overall well-being. Second, SDG 9, Industry, Innovation, and Infrastructure. This project demonstrates biotechnology-driven innovation, applying enzyme-assisted extraction to marine biomass for high-value food applications. Third, SDG 12, Responsible Consumption and Production. Valorization of the underutilized seaweed Sargassum polysystem supports sustainable resource use and reduces reliance on conventional raw materials. And fourth, SDG 14, Life Below Water. Harvesting and valorizing Sargassum polysystem contributes to the sustainable use of marine resources while addressing coastal issues caused by its overgrowth. Unlocking Sargassobiotique, ocean-derived sustainable prebiotics.